now for you?
Yes, we can see and we can hear you. That's great. So the floor is yours. I will leave you now.
Oh, great. I will do
Maybe
my
maybe just
best.
yeah,
Yeah.
just put on the whole screen the presentation. Yeah,
Okay.
present.
I am in a full screen, but I don't know why it is not sharing the full screen on your computer. We'll try something else. Sorry for the time.
No, it's okay.
Maybe like this, it is better now.
Oh, that's great. Great.
Good. Great. Thank you for giving me the opportunity to presenting our current research. I hope, oh, sorry, everything is going. It's better. So yeah, I'm Angélique Goffin. I am an associate professor at the University of Paris-Créteil. I'm working with Professor Gilles Varro, but also we are in the same lab as Régis Moiron and Julien Leroux, if you know them, for our project. Our work is more about studying how we can develop in situ and online high frequency measurement of organic matter into wastewater and river. Why we focus on this type of studying is because we want to focus on analyze organic matter. Organic matter have huge impact into the aquatic environment. First, about nutrient cycling. As we know, it can induce algal blooms. Moreover, for desoxygenation event, it can also induce fish mortality. And organic matter have also a huge influence into bioavailability of micropollutants, and it is also a major source of CO2 in atmosphere. Studying organic matter is also important as it is also playing a big key role into the wastewater treatment plant. As we know, it can impact the river quality if it's not well treated. So it is one of the main parameters to be eliminated. And it is the same for the drinking water treatment plant where we need to produce a great drinking water quality. So it is also one of the big main parameters to be eliminated. This is why we need to characterize and have a well-known information about organic matter quantity, but also its chemical nature. And if possible, if we can have this information in situ and at high frequency, it is better because you will be able to have a better understanding of the biochemical process into the river, but also having a real-time treatment plant management efficiency if you have those information very earlier than what we can do with currently used measurement method in the lab. So several methods existing for characterizing organic matter. I will not explain all of them, but usually all those methods takes time to be made because you have to prepare the sample. You have also some time to isolate some fraction of organic matter, and they need to be made at the lab. And if you want to have a fast information about organic matter quality and quantity, you have to focus on some analytical methods that are okay to be used in this type of configuration. So this is why we're focusing on using 3D fluorescence spectrometry, as it is a fast and inexpensive analytical method. It can be used in situ, in real time, and also with a high frequency measurement capability. So what it is, this method? Just very fast, this method use light. You sending light at a certain wavelength and excitation wavelength level of energy, you're sending this into the sample, and the sample will react to this light and emitting another light, which will be detected by the laboratory measuring device. And you will obtain this type of map, which is three-dimensional because you have one dimension with excitation wavelengths, another one with emission wavelengths, and the third one, it is the fluorescence intensity. The fluorescence intensity can give you various information. First, the more it is red, the more you have fluorescence intensity, and the more it's cold color, the less you have fluorescence intensity. So you can have information about the quantity of organic matter you have. But okay, you have this map, and what do you have of information with this map? You have fast information about quality also of organic matter. If you look at the back on the blue square of this picture, you can see that fluorescence intensity, it's located more in this area. So if I want to have a good understanding of this map, I can compare with literature information, and this gives me information about if I have my maximum of fluorescence intensity in this part of this map, I can have usually compounds that are generally hydrophilic and more readily biodegradable. If my maximum of intensity was more into the red square, it was more for having organic matter, which is generally hydrophobic and poorly biodegradable compounds. So this is information are really important because it can allow you to have fast information about the treatability of the water. And for example, if you have a biological treatment unit, having information very early about how it is easily biodegradable or not your organic matter is, it is very important. And for the other information you can have, for example, if you have membrane treatment, having information about how hydrophobic and hydrophilic organic matter is also a good interest. So all this project started since 10 years ago. During my PhD, yes, I'm beginning to be old now. During my PhD, we observed that biogeochemistry of dissolved organic matter can be followed by this method. And also, we also highlighted various proxy of fluorescence excitation and emission wavelengths that could be used as a proxy of biochemical oxygen demand and also chemical oxygen demand measurement into the wastewater. So we can have various information very fast about this measurement by using fluorescence spectrometry. It is the same with a new parameter we highlighted, methanogenic potential of sewage sludge can also be measured by 3D fluorescence spectrometry. Sorry. So all of these results give us a very good overview about how we can use this 3D fluorescence spectrometry to improve water treatment and to know better aquatic environments. However, all this work was first carried out by using grab sample, and grab sample is not allowing you to having high frequency. So this is why we need to transfer the laboratory measuring device to a small probe that can be used in situ and in real time. And this is the next part of this story I will present in you. So during my postdoc, we think about what it is already available into the market for doing the same as we did in the lab. But sadly, when we did this review, we doesn't really have the opportunity to highlight a very good fluorescence probe that can be very efficient to have a lot of information about various excitation and emission wavelengths, because if you want to have a very good characterization of organic matter, you have to be able to measure several excitation and emission wavelengths pair. And on the sensor available into the market, you only have 2 or 3 excitation emission wavelengths available. And the map I showed you just before, it is more than 2,000 excitation and emission wavelength pairs. So you see the gaps is very important. So by observing that, we say, okay, maybe we should develop something that could give us like a middle solution between too much information and not too much information. And this is why we developed a probe called Fluocope into the research program Mocope. So several steps happen during this development of this sensor. First, we have to choose what technological choice it is the best to build this sensor. We have to think about what detector we should use, what excitation sources we have to use, what various problem about electrical measurements we can encounter. So it is a lot of things to fix. After having various proof of concepts and prototype we made at l'ESU, we finally arrived to build a functional prototype, and it was subject to various continuous improvements. And since May 2023, we have now a first industrial probe available. The advantage of the sensor, it is able now to analyze 32 excitation emission wavelengths, which is very better than only the 2 and 3 excitation emission wavelengths usually used into other sensor. We are able to having a good quantification limit, 7 to 25 lower than the concentration measured into the river, which means we can use wind sensor in various environments. We also improve drift control system, which is not very used into various sensor of fluorescence currently. So what is this sensor? This sensor is, luckily, we have the opportunity to patent it. So we are very happy to have a patent of this sensor. But this picture showing you the last version of the sensor, you have two parts. One part, which is the part called B, surface module, which inside having a personal computer, allowing you to manage the measurement frequency, the excitation emission wavelengths you want to follow, and also you can program the you can schedule the when you want to do the measurement or not. Also, in the second part, you have the part A, which is the immersed sensor. The immersed sensor having various excitation sources, which are LED, and also it is in contact directly into the middle. So it is a very usable probe that can be used in situ and in real time, as it can do measurements each 15 minutes, which is very fast for having information about water quality. If you want to know and have a lot more of information about this sensor, we recently published a paper. You can go in to have more information about the sensibility of this device. And we compare this device to a laboratory measuring device, and we show that both of them are very good together. As I showed you before, this is you can saw at the lower part of my screen a map with white crosses. The white crosses are the excitation emission wavelengths the sensor can monitor, and the other, it's the map that we usually obtain with the laboratory measuring device. So we can see that the sensor is allowing us to manage a lot of excitation wavelengths, will give us a lot of information about organic matter quality and quantity. So now, an example of application of this sensor, we use this sensor to analyze wastewater treatment plant settling effluent by doing one measurement each 15 minutes, and we deploy this sensor into a wastewater treatment plant called Senaval. It is it is the biggest of France. It is located near to Paris and receive a lot of wastewater for the greater Paris area. In this graph, you have the fluorescence intensity into various equivalents that allowing us to see various information about the quantity of organic matter. And as you can see, by the day of the week and the day of the weekend, Thursday, Friday, and Saturday and Sunday are presenting very different shape of variation of intensity. This graph giving us very good information about significant variation of discovered organic quality and quantity over a short time, which is not really well observed with other sensor. We also have an idea to explain what what this graph is different during the weekend and not during the week. Maybe it's due to conjunction with human activity. And by knowing all those information very fast, we can adapt a reagent addition and aeration level into the biological treatment unit. So this story will be continued into two PhD, which are currently made in the lab. After using the sensor for having information about the quantity and quality of dissolved organic matter, we can also use this sensor as a proxy of various parameters which are usually measured in the lab. Like, for example, we highlighted correlation between fluorescence intensity with dissolved organic carbon, but also fecal indicator bacteria and BOD5 and COD in wastewater. So we have various applications that can be do with this sensor. One example with the dissolved organic carbon predictive model we highlighted. This is a predictive model highlighted into the river. We made the measurements into the Seine River basin with Marne, Seine, and Oise rivers, which are upstream and downstream of Paris area. And we use a huge number of samples, which allowing us to having a very good correlation between the predicted DOC by fluorescence spectrometry and the DOC concentration measured by usual method. We have a similar approach with BOD5 and COD into the Seine Aval wastewater treatment plant settling effluents during dry weather condition. We highlighted also with fluorescence intensity and BOD5 and COD measurements that we have also very good correlation with very low residual observed. But further validation of those models need to be obtained because the model I presented to you for BOD5 and COD were specified to be used into dry condition. We have now to check if it's already still available and functioning for rain weather condition. The sensor can also be used for various other possibility and problem. For example, if you want to have information for clogging phenomenon or forming phenomenon, organic matter monitoring by this sensor can give information to explain the processes. In the future, we're also going to do an economic study for having information about how this sensor can also give a good impact into the management strategy of the wastewater treatment. For example, we know into the biological treatment, you have to add sometime methanol, and also you have to increase or decrease the oxygen use. So having information about how this technology can have good impact into the treatment will be also studied soon into 2 PhD currently made in the lab. I explained to you just before having this part of this presentation more parameter related to water, but maybe we can also have use of this probe for sewage sludge management. And PhD recently finished by Maxime Dechaine bring information about how we can use, for instance, spectrometry to having biometagenic potential measurements with, for instance, spectrometry. So he compare the near-infrared spectrometry analysis usually made to analyze BMP sometime, and he compare it to filtered and diluted supernatant of sewage sludge analyzed by fluorescence spectrometry, and he also compared it to the usual method used to analyze BMP by AMPTS. On various sewage sludge from various wastewater treatment plants of various nature of the Parisian area, he obtained finally a model that can be used based on fluorescence intensity. He correlates it to the measurements made by MPTS, which are usually take 5 weeks to be made. And by comparing it by fluorescence spectrometry measurements, which are less than an hour, we can have now a proxy of measurement of this parameter of interest into fluorescence spectrometry, which is very a good improvement for having fluorescence spectrometry used for management of the sewage sludge. I speak a lot about wastewater treatment, but the river should be also highlighted. So we are going now to use the fluorocopy prop into the Seine River basin. We also are going to build a dissolved organic matter observatory, and here you can see we had and we have various sensors already implemented into various places at upstream and downstream of the Paris area. The first deployments were made in 2023, and in the next future, we are going to continue this deployment to the Seine River estuary. Two other sensors are also used for having more information about the urban contribution into the river quality. For example, we have one sensor at one wastewater treatment plant outlet and another one into a combined sewer overflow, which is one of the biggest of the Parisian conurbation area. This information will give us a lot of new input to the research field of dissolved organic matter. As we don't have into the literature a lot of similar structure of dissolved organic matter observatory. This high frequency and in situ measurements will allowing us to having a lot of information about fluorescence property of dissolved organic matter, and also having parameter of interest by predictive model I just presenting you before, like for example COD, FIB measurements. We also are going to have good information about the nature of dissolved organic matter according to hydrological situation and season, having better information about how urban discharges impact the river quality, to having a better understanding of the biochemical processes into the river. Those information will be used into a future model prediction for having a good understanding of the whole chemical processes. And finally, having information about the germ treatability is very important for the drinking water treatment plants. So to conclude this presentation, we have now a probe that can be used into various environments, into the wastewater treatment plant, into the drinking water treatment plant, also into the receiving environment, and this will allowing us to having various application. First, for BOD5 and DOC, which will be very important to have real-time high-frequency information of these parameters will be very important for improving the treatment plant. And moreover, those measurements will help to have a lot of good economy and also energy saving into various situations. So this is the end, and this is the picture of all the people into the project. So don't hesitate if you have questions and want to solicit us. We are here.
Thank you, Angélique. Now I will give the opportunity to our audience to, if they have some question for you. So would you be kind just to be here for a few minutes more?
Yes.
Do we have some questions? And also for the online listeners, if they have a question, they can also. Somebody, yeah. If somebody have the question from online listeners, you can just ask. It's okay. I have one question from here. So, Georgia, can you come here?
Okay, it's a general question, but it's regarding the Seine River quality. I just wanted to ask Kairos, I see now everybody, what is the quality now when Olympics are over? Do you see the fluctuation getting back to the old, or you continued with better treatment and better sand river quality? What's happening now, a year after almost?
So this is a very good political question about the Seine River quality, but what we know, it has never been so much good now than before. It's always improving the water quality because a lot of efforts was made to improving the management of the sewer overflow. Less sewer overflow contribute to increase the Seine River water quality, but they also had a lot of water treatment good the water treatment management were improved into other wastewater treatment plants, but now it's never be as good as it's good now. It's better now than in the past, and it's continue to improving the quality now. Yeah.
That's great. Thank you very much. Do we have any more questions? No. Thank you, Angélique, very much for giving this interesting lecture. I will now continue on Serbian, don't mind.
That's okay, don't worry. Thanks.
Thank you very much. Dakle, ovde sa ovom prezentacijom bi zapravo završili ovaj zvanični deo skupa. Sada nas očekuje pauza za ručak i ono što je izuzetno bitno, ko se prijavio za radionicu nakon ručka, naša koleginica iz IKRE, Laura Bertolini, će nas u stvari uvesti u priču pripremanja projektnih predloga. Dakle, memorijalni naučni skup nastavit će da okuplja i dalje mlade istraživače u uverenju da budućnost ne čeka, već da se aktivno gradi. Kroz nauku, debatu i angažman mladi oblikuju ne samo svoja istraživanja, već i pravac kojima društvo ide. Zato je važno da nastavimo da negujemo mesta poput ovog skupa, gde se slobodno razmenjuju ideje, gde se podržava razvoj i gde mladi dobijaju priliku da budu ono što i jesu, pokretači promene. Dakle, ručak će nas opet sačekati u salonima. Nadamo se da će biti promena. Ručak će biti u salonu, nakon ručka nastavljamo za one koji su se prijavili kroz radionicu. Hvala puno. Thank you very much, Julienne and Angélique. See you in France, hopefully this summer.